Hey, good afternoon, everyone, and welcome to the 2013 Fall TCAP webinar series. This afternoon, we have with us Dr. Rex Bernardo from the University of Minnesota. Dr. Bernardo is a professor and endowed chair in corn breeding and genetics at the University of Minnesota. He obtained his bachelor's degree in agriculture in the Philippines and his PhD in plant breeding at the University of Illinois. Prior to his current academic position, he was formerly a research scientist with lemur grain genetics and professor at Purdue University. At Minnesota, Dr. Bernardo conducts research on new ways to breeding corn and breeding corn for new uses. He also teaches courses in plant breeding and scientific writing. He is the author of a textbook entitled Breeding for Quantitative Traits in Plants and is currently writing a prequel entitled A Primer of Plant Breeding. Today, Dr. Bernardo will review the basic of genomic selection and will share results and ideas on how to routinely implement genome-wide selection in an inbred development program. Let's all welcome Dr. Rex Bernardo.
Thank you. Can you hear me well?
Yes,
All
yes, we can
right.
hear you.
I'll get started. I apologize for the technical difficulties we've had over here, but I think we have that figured out, so we're going to go ahead and get started. So it is a pleasure for me to speak with you this afternoon to share about thoughts on how to routinely use genome-wide selection in an inbred development program. It's not too often I get to talk to wheat and barley people, although I do have a wheat and barley breeder down the hall, so it's a pleasure to join you this afternoon. I'm not using PowerPoint slides, but I'll be writing a few things down on sheets of paper. And so to get started, what I'd like to do is tell you what I'm going to talk about this afternoon. And what I'm going to talk about could be summarized in three main points. Number one is don't forget major QTL. As we begin to implement genome-wide selection routinely in a breeding program, I think there's a danger of focusing solely on genome-wide markers to the extent that we don't remember that there are major QTL for different traits, and we need to bring those along in the breeding process. A second thing we've found is that you can't predict the prediction accuracy. In other words, we want some assurance as we start the experiment that the prediction accuracy will be high. However, it turns out that doing so is quite difficult, and we might just need to throw up our hands and basically say, in the same way that we cannot predict heritability in advance, we cannot predict prediction accuracy in advance, although there are best practices that we could follow to try to ensure that prediction accuracy will be high as possible. A third lesson that I'd like to talk about is that in genome-wide selection, we often think about the composition or the size or the number of markers in the training population. What I propose to you today is our first order of the day should actually be the test population. We should first determine what the test population should be, and that the training population is only secondary. That is, if we want to apply genome-wide selection routinely in a breeding program. So to introduce this topic, what I'd like to do, as Jayford indicated, was have just a very brief review of what genome-wide selection is all about. And to do that, I'd like to draw on a hypothetical yet simple example. I found that there is this species called jumper jack ant that only has one pair of chromosomes. So it has the minimum number of chromosomes in any species. And let us say that we have here 8 ants with different body lengths from 12.1 millimeters to 9.8 millimeters. And on this one chromosome, we have analyzed 3 different markers, 1, 2, and 3, and we are assuming that the markers are homozygous. So we're coding them as 1, -1, and we're just assuming that there are no heterozygotes or zeros for now. Now, if these are our data, we are all quite familiar with multiple regression, in which the model for multiple regression is y sub j. is equal to the slope, b sub o, plus the effect of marker 1 times the indicator for marker 1, plus the effect of marker 2 times the indicator for marker 2, plus the effect for marker 3 times the indicator for marker 3. So again, nothing too complicated. It is our basic multiple regression linear equation. And so if we try to solve what the regression coefficients are in this example, what I did was I tried to construct this in a way so that marker 1 and marker 2 and marker 3 are independent from each other. And you see that because of this alternating 1, 1, 1, 1, 1, minus 1, and so on. And because of that independence, we can easily solve the regression coefficients as follows.
Rex, excuse me.
Yep.
Sorry to interrupt. Some of the participants are having problems with static in your audio. Could you turn minimize the volume of your microphone?
Okay.
Is that right? Yeah. Sorry.
Yep. No problem. If I can figure that out, is that better? Can you still hear me?
Yeah.
Okay. Is it a bit better now?
Yeah, that's better.
Okay.
Okay. Thank you.
I'm just speaking loudly today, I guess. Too much coffee. Too much coffee. Anyway, so in this special example, the three markers are independent, and then we can solve for the regression coefficients as follows. For example, the slope, in this case, is simply equal to the mean. It's a summation of yj divided by the number of observations, which is n. So the slope is equal to 12.1 plus 11.9 plus 11.2 and so on, and that gives us 88 divided by 8 observations, and that gives us our slope as equal to 11, and that's equal simply to the mean. So nothing too complicated. We can also obtain the regression coefficients according to this formula, summation of xj, yj, that means to say that is the sum of cross products divided by the sum of squares of x, meaning that we multiply 12.1 times 1 plus 11.9 times 1 plus 11.2 times 1 plus 11 times 1 minus, plus 10.8 times minus 1 and so on. So basically what we're doing is we're getting the sum of the observations with plus and the sum of the observations with a minus and dividing it by 8. So that gives us for B1, I get that as 4.4 divided by 8, and that gives me 0.55. If I do that for B2 and for B3, the same thing, sum of cross products divided by the sum of squares, that gives me 3.6 divided by 8, 0.45, and finally B3 is equal to 0.6 divided by 8, that's 0.075. And basically what we're saying then is that these regression coefficients are simply equal to the slope. It is the change in y, delta y, over the change in x, as the markers go from minus 1 to plus 1. So again, quite a very simple example. Now, a common method that has been used in genome-wide selection, as we know, is called ridge regression, best linear unbiased prediction, or RR-BLUP. In multiple regression, we assume that the effects of the markers are fixed. In ridge regression BLUP, though, we assume that the effects of markers are random, and what happens is that there is shrinkage of the effects towards the mean. And in fact, in ridge regression BLUP, in the context of genome-wide selection, we'll see that the larger the number of markers, the greater the shrinkage is towards the mean. Now, those of you who are familiar with ridge regression BLUP know that the variance of marker effects is equal to the total genetic variance divided by the number of markers. That is to say that if we have 100 markers in the model that we are constraining each of the markers to account for 1% of the variation. If there are 200 markers, we are asking each marker to account for 1/2 of 1% of the variation. And it turns out that the shrinkage factor can be nicely written according to the following form. The VE, the residual variance divided by the variance of the markers, is simply equal to the number of markers times 1 minus heritability divided by heritability. And the nice property is, as we know, heritability ranges from 0 to 1, which means that we really do not need to know what the actual values of VE and VM. Instead, what we need to know is the heritability of the trait. And so because of that nice property, we don't need to write down VE or VM in this equation here for shrinkage. And when we do that then, when we compare the marker effects from ridge regression block with the marker effects from multiple regression, this is what we get. The ridge regression block marker effects, by the way, can be obtained using this equation only, and only in this special example we have where the markers are independent. When the markers are not independent, when they are linked or are in linkage equilibrium, then this formula cannot be used, but this formula is useful for illustrating the property of ridge regression block. If you remember, this first numerator here is the same as in multiple regression, and this first part of the denominator is also what we have in multiple regression. What we're doing then in ridge regression block is that we are adding a certain term to the denominator. It makes the denominator larger, and it causes the value of BI then to be smaller, meaning it is shrunk towards the mean. And we see here also that the larger the number of markers, the greater the shrinkage would be, but the larger the heritability, the less the shrinkage would be. And so what we could do then is, based on this formula, determine what the marker effects would be for different levels of heritability. For example, in the multiple regression model that we worked out, the regression coefficients were 0.55, 0.45, and 0.075. If heritability is 0.75, the marker effects become 0.49, 0.40, and 0.067. So you see again shrinkage towards 0. If heritability is even lower, 0.25, the estimates of marker effects become shrunk further to 0.26, 0.21, and 0.035. And so we see then, obviously, that if heritability is 0, then those effects would be further shrunk until they become equal to 0, which is what we should expect if heritability is indeed 0. And so I hope that this here nicely demonstrates what happens, how ridge regression block estimates of marker effects are related to multiple regression estimates of marker effects. So now when we apply this to the prediction process, suppose now that we have two ants, numbers 9 and 10, that have been genotyped but not phenotyped, and let's say that heritability is 0.75, and we want now to predict the body length of ant number 9. So at 0.75, the marker effects were 0.49, 0.40, and 0.067. That means that the body length of ant number 9 is 1 times 0.49 plus 1 times 0.4 plus 1 times 0.067, and that gives us 0.96. Now for ant number 10, it'll be 1 times 0.49 plus 0 times 0.4 minus 1 times 0.067, and so the predicted value length of ant number 2 is 0.42. That is ridge regression block. What if, however, what if marker 1 is a major gene? What if marker 1 is a major gene, and instead of treating it as a random effect in ridge regression block, we now treat it as a fixed effect, because we know it to be a major gene. What happens then, if the multiple regression estimate with for marker 1 was 0.55 if the marker effect was fixed. And so what we have now, if marker 1 is for a major gene, is that the predicted body length of ant number 9 becomes 0.55 times 1 plus 0.4 times 1 plus 1 times 0.067, it increases to 1.02. And if we do the same for ant number 10, this increases from 0.42 to 0.48. So we see here then that it does make a difference if our markers have a fixed effect or a random effect. And from talking with colleagues here in wheat breeding and genetics, I know that in wheat, for example, there are major QTL that have been identified. For example, the FHB1 is a major QTL for scab. There are glutenin genes that affect dough quality. And so a question then is, well, if I am selecting for both FHB1 as well as polygenic effects for scab, or if I am selecting for the known glutenin genes and polygenic effects for dough quality, what effect what would be the effect if I then give special treatment to these known genes or QTL and assume that they have fixed effects? So what I did was then I conducted a simulation study in which I considered the effects of having L equals 1, 2, 3, or 10 known major genes. In addition, I considered that there were 100 background QTL affecting the trait. Background QTL had different effects. And then the R-squared value, or the proportion of the phenotypic variance accounted for the QTL, ranged from 5% to 50% when there was only one major gene affecting the trait, or when there were 2, 3, or 10 major genes, the R-squared, I assumed, was equal to 50%. And so the question then was, in this model, by the way, I had 438 SNP markers, and this was for my favorite species, which is maize. Sorry about that, but that's okay. Principles remain the same. So the question, the basic premise is, let's say that one of these 438 markers are for one major gene. Would there be a difference if we assume that all 438 have random effects, or if the 437 have random effects, and then the one for the major gene has a fixed effect? And the same idea if there were 2, 3, or 10 major QTL or genes controlling the trait. What is the effect basically of assuming or of giving special treatment to these known genes? And this is what I found. This is the relative efficiency or the response to selection when we consider a fixed versus random effect for a single major gene. This is after 4 cycles of selection. And what we have here is different population sizes, 250 lines or individuals versus 100. Different R-squared values for the effect of the major gene from 50, 33, 25, 17, 10, and 5. Different levels of heritability for the trait. And here's what happens. If the population size is large, if the R-squared is high, and if heritability is high, there is a 12% advantage. 12% advantage in assuming that the major gene has a fixed effect. as we would expect, as the R-squared value for the major gene decreased, the advantage of assuming a fixed effect for the single major gene also decreased, although they were always significant when heritability was 0.8 and the population size was 250. We see here that when heritability decreased to 0.5, there was still an advantage of assuming a fixed effect for the major gene when the major gene had a large effect, say 0.25% R squared, but not with a small effect. And we see here, though, that in a way heritability trumps everything. If heritability is low, in this case 0.2, it did not matter in terms of selection response if we assumed a fixed effect or a random effect for the known single major gene. What we see here also, though, is that when the population size was small, when the R squared was low, there was actually a disadvantage to treating the single major gene as having a fixed effect. We would have been better off ignoring, in that case, ignoring the identity of that known major gene if its effect was small and population size was low. Overall, though, what we see here is that, for the most part, if R squared is equal to 10% or more, treating the major gene as having a fixed effect did not do any harm, meaning that we did not always have an advantage in assuming a fixed effect for the known major gene, but at least it did not do any harm except for this one exceptional case. So the take-home lesson here is that, yes, when we implement genome-wide selection routinely in an inbred development program, do not forget major genes. And if major genes are known, we need to treat their effects as being fixed as opposed to being random. So that is the first lesson, I believe, one lesson that we've learned in using molecular markers routinely in a breeding program. Now, shifting gears a little bit, a second lesson that we have learned is that as much as we want to predict the prediction accuracy in advance, it is extremely difficult to do so. By that I mean, let's have a few definitions here. Let me define two terms. One is RMG. This is the correlation between the marker predicted effects, the marker predicted performance, let's say, and the true genotypic value, or the true performance, which is not known, of course, unless it is a simulation experiment. What we can actually estimate in real life, directly at least, is RMP, which is the correlation between the marker, the performance predicted from markers, and the phenotypic value, which is what we observe, but at the end of the day isn't really what we want. We're after the genotypic value. So in the rest of my presentation, I'll be referring mostly to RMG, although I might refer from time to time to I'm sorry, I take that back. I'll be referring to both correlations later on. Now, there was a study by Detweiler. There was a study by Detweiler in 2008 and 2010. In which Detweiler and colleagues derived the expected prediction accuracy. And they derived it as follows: that the expected prediction accuracy, the expectation of R and G, is equal to the population size, which is n, times heritability, divided by the population size times heritability, times the effective number of chromosome segments, which I'll define, and you take the square root of that. That, again, is the theoretical accuracy of prediction. It's a function, as population size increases, the prediction accuracy increases. As heritability increases, the prediction accuracy increases. However, it is not, those two terms are multiplied, meaning that a larger population size can compensate for a lower heritability. And if heritability is low, you do not need as large a population. ME is basically the effective number of chromosome segments. It also has been defined as the effective number of QTL. It's an idealized situation. It is the equivalent number of QTL if the QTL were behaving in an entirely additive manner, unlinked to each other, and they had equal effects. So those of you who have taken quantitative genetics and are familiar with the concept of effective population size, it's a sort of similar kind of analogy with the effective number of chromosome segments. And so what a former student of mine did, Emily Combs, was she studied, well, what if we keep N and H and the number of markers constant? Will different traits have the same prediction accuracy if we keep all things constant? And she did that by adding different amounts of noise to the level of heritability. For example, if heritability of trait 1 was equal to 0.50 and the heritability of trait 2 was equal to 0.60, what Emily did was add the right amount of noise to the phenotypic data so that this heritability would be equal to 0.50 instead of 0.60. And this is part of what Emily found. Emily, this is for the same population. Again, the same barley population, the prediction accuracy, heritability, same number of markers, same N, and different traits. And what we see here is that even if the population size is kept constant, even if heritability is kept constant, even if the level of LD is kept constant because it is the same population, different traits differ or vary in their prediction accuracy. Some traits are just more predictable than others. Extract here has a low predictability. Height has a high predictability, and I don't think that's any surprise. And I think this is then entirely consistent with the fact that some traits just have a high heritability, some traits have a low heritability, and we do not exactly know the reason why. And the same again, some traits are easy to predict, some traits are difficult to predict, and we don't know exactly the reason why, and so we need empirical evidence or experience to determine which are the traits that are more predictable and which are the traits that are less predictable. Now, this we also see that here. This is the work by another student, Lian, in my program. We were fortunate to be given access to data from Monsanto Company for 1,000 biparental populations in maize. And that's not a typo, it was 1,000 populations, so we were very thankful for that. And what we have here is the prediction accuracy in RMG for different traits. And we see from these box plots, yes, that indeed the mean prediction accuracy varies, but then there is also quite a bit of variation in the prediction accuracy. The last column here, for example, is yield. The prediction accuracy was about 0.25 mean. However, there's quite a bit of variation in the quartiles that you see plotted in here. And so again, in general, we can say which traits are more predictable than others, but overall, there is quite a bit of variation in the different prediction accuracies that we get. Now, this formula here by Detweiler, by the way, assumes that we have total genome saturation, that the number of markers is very high, so that the genome is entirely saturated. And that wasn't the case in our our data. So we modified this formula to account for the recombination frequency among the markers. I won't bother putting the formula, just again, it just includes an extra term, an extra parameter that is a function of the heritability. And what we found was, this is for the 1,000 data points and the different different traits. On the left is the Detweiler equation, and on the right is our equation. And what we have here plotted is the expected prediction accuracy on the x-axis and the observed prediction accuracy on the y-axis. And we see here, this line is the unity line, and so the Detweiler formula overestimates the prediction accuracy. Our formula does a better job at predicting the mean of the prediction accuracy, and we see though that there is quite a cloud, still a cloud of data points about that mean. And so again, it goes back to our conclusion that it is extremely difficult to predict prediction accuracy. However, I don't think that everything is lost, because as you remember, if you remember that formula by Detweiler, a key parameter is the product of population size and heritability. And you might remember as well that n times h squared is a key parameter that determines the power to detect QTL in QTL mapping experiments. So what we did was we tried to plot the prediction accuracy against nh squared, and this is what we obtained. We found that so nh squared is on the x-axis and the prediction accuracy is on the y-axis. And we haven't spent enough time looking at this plot, but it doesn't take too long to see that there seems to be quite a distinct triangle of points over here. Now there are a few points below this triangle, but for the most part, most of the data points are above that triangle, which means to say that it seems that we might be able to specify rules of thumb for us to follow with regards to nh squared. For example, when nh squared was equal to 100, then the prediction accuracy was about 0.25 for the most part. If it was 125, then the prediction accuracy was about 0.5 and higher for the most part. So again, it seems that although we cannot predict prediction accuracy in advance, we could set some rules of thumb simply based on the product of n and h squared, and of course that is assuming that we have enough markers to saturate the genome. So again, the second lesson, first lesson was don't forget major QTL. Second lesson is we cannot predict prediction accuracy. And the third point that I'd like to share as we consider using genome-wide selection routinely in a breeding program is that we need to think about the test population first and the training population second. Before I go into that, I'd like to talk about three terms that I've already used. We're talking about the training population. We have the test population. And you may also have heard of the term validation population. So what I'm talking about here is that the training population is used to develop the prediction model. Okay, we use the training population to develop the prediction model, and then to see how well that works, we apply that to a validation population, again to see how well this prediction model works. When we begin to apply the prediction equation routinely, then we use this prediction equation to predict the performance of a test population. And as we do that, when we predict the performance of the test population, what might have been the validation population might be pooled with the training population, so that we just have one large training population that is used to predict the performance of a test population. If we are to apply genome-wide selection in a breeding program, we need to carefully consider the breeding context in which those predictions will be used and to consider how breeding is currently done. For example, in wheat and in barley, I would surmise that most of the breeding is done by crossing two inbreds, A by B. You get your F2, you get your F3, and then you get a bunch of recombinant inbreds. You'd repeat the same process with two more parents, say cross C by D. You get the F2, you get the F3, and you get recombinant inbreds. And you could have a third and fourth and fifth. Let's write one third, E by F. You get the F2, you get the F3, and you get your recombinant inbreds. A key question that I believe needs to be asked is, how does the breeder do the selection? For example, does the breeder select within each recombinant inbred population independently? By that I mean that the breeder would select the best recombinant inbreds in A by B, go on to the next population, select the best inbreds in C by D, and then go on to the next population and select the best inbreds in E by F. In that case, A by B, for the purposes of genome-wide selection, A by B is one test population, C by D is a second test population, E by F is a third test population. Is that the way the breeder operates, or does the breeder make selections among all of these individuals pooled together? Meaning that in looking at the data, the breeder will pool all of these recombinant inbreds together and simply select the best without regard to which population the inbreds come from. In that case, the test population is this pool of 3 biparental crosses instead of 1 biparental cross at a time. Or is the breeder going to, so by the way, this then typically would go into, say, year 1 testing. Or does the breeder then select the best in population 1, okay, the best in population 2, and the best in population 3, and then put all of these best individuals in one yield trial in year 2? In this case, the test population, the test population is this pool of individuals here from 3 different populations, but where selection has already been done. And the point here that I'm trying to make, again, is that depending on which system of selection we'll be using, we might want to use different kinds of training populations. Specifically, if the test population is sort of like a panel of different inbreds, inbreds from cross number 1, inbred from cross number 2, inbred from cross number 10, some inbreds that might have exotic germplasm, and so on, then it seems that an appropriate training population should also be in the form of a panel. However, if we are trying to select among recombinant inbreds within, just within a cross, such as A by B, this means that we should not use a panel as our training population because a panel might not predict performance within a specific biparental cross. And the reason for that is as follows. For example, if we try to plot, let's say this is, let me make a graph over here, where this is the observed performance, and this is predicted performance. What we may have is the recombinant inbreds from the cross A by B, having that distribution. We could have recombinant inbreds from C by D, and then we might have, for example, recombinant inbreds from E by F. What that means to say is that if we pool all of these crosses together, we can surely fit a nice straight line, and we say that our prediction accuracy is pretty high. What we quickly see, though, is that that high prediction accuracy is largely due to the differences among the means of these different biparental crosses as opposed to the differences within each biparental cross. Meaning that if we are now applying that prediction equation to just A by B, then we won't have very good performance of the prediction equation. Meaning that, yes, if we have a diverse panel in our test population, we should use a diverse panel for our training population. If we have selection within recombinant inbred lines, what we're finding out, and I can't share too many results today because they're still under analysis by one of my students. But what we're finding out, though, is that if we are interested in predicting the performance in a cross between two inbreds, A by B, that a good training population to use is all the inbreds from the cross of what I would call A by star and B by star. Meaning that we go back to our data, we try to find all the populations that have used A as one of the parents. We find all of the biparental populations that have used B as one of the parents, and then we pool all of these together into one training population. And so we see there's a half-sim structure then between the test population and the training population. And so again, I don't have many results to share of this specific scheme right now because they're still under analysis. But the main point, again, that I would like to emphasize is that we first need to think about what is the test population. That's our main priority. Once we have figured out what the test population is, then we try to figure out what is our training population, as opposed to the other way around, which in many cases does not work well. So with that, I'd like to conclude my talk, and thank you very much for your attention this afternoon. If there are questions and if we have time, I'd be happy to try to answer them. Thank you very much.
Thank you, Rex. I think we have a first question here from Bao Honggu. Going back to your first topic about the first part, he was asking the question. So you suggested to propose a linear mixed model by treating the major QTL as fixed. However, generally speaking, major QTLs have low frequency. So he suggested that what if to do marker selection first and do genomic selection after. So I don't know if you have any information.
Yes. So one possible alternative is to first use markers to screen for the major QTL, and then to use after that has been done, it's to then use genome-wide markers in a genome-wide prediction equation. My opinion on that is that genome-wide selection is a form of index selection. It's a form of index selection in the sense that you give different weights to different pieces of information. And it has been found in the literature that index selection for improving multiple traits is superior to tandem selection. So if we just talk about improving one trait at a time, instead of improving trait 1, then trait 2, then trait 3, it has been firmly established that index selection is superior. And from that standpoint, that is why I simply propose to use more of a selection indexed kind of approach where we consider both the major QTL and the background markers at the same time. I do appreciate the point that if it is a major QTL and if it is in a low frequency, then that might pose some problems. In that case, however, there might be some prior information available on that major QTL. For example, there could be prior information on the mean effect of the major QTL, and perhaps that such prior information could be incorporated in the selection process.
Okay, thank you. Is there any more questions from participants? You can just type into the chat box and then I'll ask Rex. Okay, I can see some people typing their questions, so. So while we wait for the questions to pop up, just based some basic questions, Rex. Going back to again to the first part where you compared a large population to a small population, or just
Mhm.
a very basic question. How did you determine that 100 is a small or 250 is a large population?
Well, yeah, that was what is small and what is large. There is obviously arbitrary. It was just comparing the two, and I'm talking more in maize. Yeah, and even some maize breeders would argue that 100 is actually large in maize breeding. So again, it was just relative to each other.
Okay, thank you. There's a question here. What if we remove the mean difference among these parental populations? Can we combine populations to build a training pool?
I think you could. So you're talking about here, if you remove the mean, if you remove the effect of the mean, then what would happen is that you have C by D over here, and then A by B would just overlap C by D, and E by F would just overlap C by D. So you don't have these two anymore, but what you still might have here is uh, it's not a good relationship. I mean, it's not a good relationship between observed and predicted performance. I think what is happening, is that when when we want to predict within a biparental cross, say A by B, first of all, that is a very narrow gene pool. It's just a cross between two inbreds. And second is that the variation is due, obviously, to recombination in that cross. And so therefore, to be able to sort out the differences in the mean that are due to recombination, the type of training population that we must use also must exhibit a high amount of recombination. And that is what we see here when we pool these different biparental crosses that share a common parent, is that we're taking advantage of the power of recombination in each of these biparental crosses. And that might not necessarily happen. I mean, that happens, of course, in a panel, but again, it's a gene space, if you would, or the diversity is so much beyond what we get within a given biparental cross. To the extent that we're back here, we can draw a nice straight line across a diverse pool, but within a smaller pool, then we don't have that nice linear relationship anymore.
Okay. I think somewhat related question. He's asking what training set do you suggest for a specific cross?
Um, it depends. If I am selecting, if I'm selecting in A by B, then this is my training data set. If I am selecting among diverse inbreds, then I would suggest a training set that is also a bunch of diverse inbreds. So again, it depends, and it goes back to the point that the test population drives the training population, and the training population should be as similar as possible to the test population that we have. Yes, indeed. It's again, I'm sorry, I forgot to mention that. We need that high degree of genetic similarity between the training and the test population.
Okay, I think we could entertain a couple more questions. Another one is he's asking with the next generation markers that is widely used now, there's a possibility of overrepresentation in portions of the genome. How can we control that in estimating breeding values if we do not have information on the positions of these markers?
All right. Well, my answer to that would be, again, I'm sounding like just a repeated recording here, depends on the training population, the test population. If my test population is the cross between two inbreds, A by B, the degree of linkage disequilibrium in a biparental cross is quite high, to the extent that there does not seem to be uh, uh, much advantage in using markers that are spaced more than less than 5 centimorgans apart. It seems that a spacing of 5 to 10 centimorgans is already sufficient for a biparental cross, and so we really don't need that many markers if all we're wanting to do is predict performance in the cross. If we're predicting in a panel, a very diverse panel, if that's what the breeder is doing, then yes, we might want more markers. And the question as to number of markers, it is difficult to answer, and I think that is best answered simply by doing cross-validation tests. If you have a certain number of markers, then you could try what happens if I use this many or that many, and then figure out what would give the highest, the best predictions in a validation population. So again, it's a more of an ad hoc answer where, given the data set, you could just figure out, we have the tools to do so, what the appropriate number of markers should be.
Okay, any more questions? Probably one more before we end. Okay, let's have this. What is the appropriate size of the training population, and how often do you need to update the prediction model?
Appropriate size of the training population, well, some would say as large as possible, and I wouldn't disagree. However, again, the answer depends, because a key parameter is NH squared, meaning that if H squared is high, I would need a lower N to achieve the same accuracy. If H squared is low, then I need to compensate it with a higher N to achieve a certain accuracy. And Emily Combs found this. It was quite a nice result. For example, if you increased heritability by 50% and increased population size to compensate by 50%, then the prediction accuracy remained constant. And so in that sense, there's no fixed answer. From a practical breeding standpoint, though, we know that heritability varies among traits. And so the appropriate N, if you would really differ, and the breeder just needs to use judgment as to what population size needs to be used. Along those lines, I'd like to comment that I personally believe that we pay too much attention to prediction accuracy. I'll say that again because that could sound controversial. I think we pay too much attention to prediction accuracy in the sense that when we do phenotypic selection, when we grow our plots out in the field, say 3 locations, 4, 5 locations, we really don't know in advance what heritability should be. In the same way, I don't think that we should worry too much what the prediction accuracy should be. A breeder, for example, would not probably grow a yield trial at one location. At the same time, we probably would have some sort of minimum population size. But I think once we determine the rules of thumb that we need to use in designing a training population, that we should just go ahead and use the procedure and not worry too much about prediction accuracy.
All right, so there you have it. I think it's about time that we end the webinar. And once again, thank you, Rex, for talking to us today. It was very, very informative, and it's the first time we have this kind of presentation.
Well, thank you. Thank you so much.
Thank you, everyone, for attending the webinar, and let's meet again next webinar in November.